Yay. Yay, OJ time. Hi, guys. It's Kathy. I hope you're doing well. We're talking about adenine, thymine, guanine, cytosine. It's going to be a good day. It's going to be a good day. Speed is already at 1.25. I'm getting really good at that. Really good. It is Saturday, right? So, we have nothing to do today other than talk about OJ Simpson. Oops. I guess we could talk about other things. Kinda.
It can be made to be single-stranded. It occasionally, in the process of making two cells out of a single cell, will exist for short stretches in single-stranded form, but that's, again, something we really don't need to be worried about. But experimentally, you can take a piece of DNA and either treat it with heat, which would be the common method, and if you think of this as a zipper, the two strands will simply come apart, so that you would have one strand that had a G, A, G, T, C, and the other strand would have a C, T, C, A, G. So it's like unzipping the zipper. You can do that chemically, you can do it with heat, and because of the specific nature of the base pairing, you can also bring it back together. The two single strands will come back together as long as each G is paired with a C and each A is paired with a T. So it's like having that zipper have four different shaped teeth, and the only way you can zip it up is if all the teeth mesh together properly, an A with a T and a G with a C.
All right, Your Honor, I was going to have the witness start with a new drawing. Would the court prefer that now, or?
No, we need to take a break for a juror issue. All right, ladies and gentlemen, please remember all my admonitions to you. Do not discuss the case amongst yourselves. Don't form any opinions about the case. Do not conduct any deliberations until the matter has been submitted to you. Do not allow anybody to communicate with you, and we'll stand in recess for 15 minutes. All right, and Mr. Clark, in the interim, would you show the exhibits, please? No, right there. Right there's fine. All right, thank you, Mr. Furlong. All right, let's have the jury, please.
Dr. Cotton, with regard to this diagram that's on the easel at the moment, People's Exhibit 237, you described how that shows DNA in a double-stranded form, correct?
That's right.
All right. Could you, with the help of a new diagram, or actually, would it assist in your describing DNA in a single-stranded condition to use and draw on what I believe would be People's Exhibit 238, a new chart, new diagram, rather?
Yes, it would.
All right. Then with the court's permission, would you go ahead and use the drawing pad again to illustrate this single-stranded versus double-stranded DNA?
All I'm basically going to do is straighten out the helix to make a second diagram that's a little bit easier for me to draw. So if we make two strands, and now all I've done is really just straighten this out so that when you look at the bases, if you have an A on one side, you'll have a T on the other. If you have a G on one side, you'll have a C on the other. And there is a bonding between the A and the T and the G and the C that we're not going to bother to draw, but there's force that is keeping these together. So you could make up any sequence here, T, A, C, C, G, for example. And if I have made up this sequence on this side of the double stranded, then I would automatically know I have a T on this side, I must have an A on the other. I have an A on this side, I must have a T on the other, and so forth. I have a C on this side, so I will have a G on the other strand, and a G on this side, so I must have a C on the other. And again, you can think about the size of the piece of the DNA that I've drawn by counting the bases. Here I have 7, so this piece of DNA that I've illustrated is 7 base pairs long. Now, if we take it apart, we haven't really left a lot of room here, but we'll just put up here, this is double-stranded. I think we better.
Go to page. All right. And if we could make that 59, thank you.
I'm not going to be able to remember the same series of letters that I wrote down on the previous one. But if I took that piece that was double-stranded and I heated it, strands would come apart. So I would have nothing attached to this side, and I would have nothing attached to the other side. However,
I'm sorry, when you say you heat it, what type of temperatures are you talking about?
you have to heat it to about 95 or 100 degrees in order to get the two strands to come apart. And there are some temperatures, some strands of DNA, if you had a lot of C's and G's, you have to heat those a little, you have to heat that a little bit more than if you have a lot of A's and T's. But basically, if you think of it as heating to 100 degrees, and when I say 100 degrees, I'm talking 100 degrees centigrade.
So not 100 degrees Fahrenheit.
Not,
So you're talking about something that's very hot in terms of a Fahrenheit measurement.
yes,
In other words, not body temperature or not a hot day out in the desert, something much hotter.
much hotter.
All right, go ahead.
If you wanted to put these back together, you can see that if you just move them back together, you were able to do that, they would all be correctly paired, and thus they could come back together and zip up. However, if I took this strand, say, and moved it down so that I had an A, or I was trying to put a T back together with this C, and an A back together with this C, and a G back together with this G, it would not come back together like that. It will only come back together when the A's and T's and G's and C's are properly paired up.
So therefore, DNA is at its happiest. Would that be correct to say when the pairs match up correctly?
Yes, it's happy and it's stable when they're paired up correctly. It's pretty happy when they're completely separate, but not as happy as when they're together.
As far as this ability to separate DNA and to have it return to its double-stranded form, why is that important?
It becomes important in the process of doing DNA testing and for many other reasons, because it allows you, suppose I have this DNA sequence, single-stranded, and I have this one in my hand in the test tube, and I mix them together. They will come back together. If I have some kind of a tag on here, some kind of tag, and the tag could be a radioactive tag or other types of tags. If I have this piece in a test tube and I have this piece in some other form and I mix them together and I let them come back together, they will zip up together correctly given some time. And now I can find out where this single-stranded piece has gone to by looking at whatever kind of tag I have stuck to it.
And is the ability to do that, or does that ability play a role in DNA typing itself?
It plays a role in every form of DNA typing that's currently produced.
Okay, we'll talk about that a little bit later. Now, as far as this overall concept of what you've described, do we in fact, to your knowledge, have a diagram that describes where DNA is found?
Yes.
Okay, with the court's permission then, I'd like to have marked as People's Exhibit 240, a diagram entitled Where is DNA Found? All right, Dr. Cotton, we've had marked as People's Exhibit 240, a large diagram entitled Where is DNA Found? Can you tell us what that diagram shows? Sure. Why don't we move the... In fact, would you be able to discuss this diagram with or without referring to this particular chart that you just made? Okay. All right, Dr. Cotton, if you could describe what People's Exhibit 240 shows.
This exhibit is basically a summary of the things that I drew earlier on the chart. So you have an example or a diagram of a cell that has a nucleus and a cytoplasm. And in the nucleus, there are chromosomes. Now, they don't have 46 of them drawn out here, but they have several, and the real number would be 46. You take one of those chromosomes out and unwind the DNA. This is a depiction of the DNA that's packaged up into the chromosomes, and if we make it larger and look at it still magnified yet further, you see again the double helix with the AT and GC base pairs.
So then does this chart, Where is DNA Found, then basically take step by showing with greater magnification each portion, or actually each way DNA is packaged down ultimately to the individual base pairs themselves?
Yes.
Okay. As far as this DNA molecule, and you've described it being the same basically in every cell that has a nucleus in our bodies, does that mean all of us are the same?
No.
Can you describe
Shall I-
that further?
shall I-
Yes, and if you would, have a seat.
Well, it's pretty obvious that we're definitely not all the same. All you have to do is look around the room and you'll pick that out right away. So within any given individual, in all of my cells, the DNA will be exactly the same from one cell to the next. And that would be true for each one of us. But from one individual to the next, the DNA will be different. Now, that isn't to mean that, we'll use Mr. Clark as an example. All the DNA in all of his cells is the same in each cell. That is, if you look at a liver cell and a blood cell, the DNA in the liver cell will be exactly the same as the DNA in the blood cell. His DNA will be different than anyone else's in the courtroom, but that's an overall difference. The great majority of the DNA, greater than 99% of the DNA, is going to be the same from person to person. And that's because we're all human beings. Our basic body structure is all the same. We all have a liver, we all have hair, we all have a kidney, and so forth. So all of that DNA information that creates those things that we have in common will be basically the same from one person to the next. There is then a small portion of the DNA that is different from person to person. And that's intuitively obvious because you can look around even in a very large room or many people, and you'll see that with the exception of identical twins, people are different, even closely related people. Two brothers are different. You can always tell them apart. And the exception is identical twins, and identical twins are identical because they have identical DNA. And that is the only exception.
With regard to, then, these portions of DNA where we differ, and I believe you said that's something less than 1% where we differ from one another.
That's right.
Why is that of interest? Why do you as a scientist care about that region or those regions where we're different?
Oh,
Many
geez,
scientists
Kelly.
would not
Breathe. Breathe. Gosh.
care about those
Scary.
regions that are different. But if you are interested in being able to distinguish one human being from the next, then it doesn't help you to look at the parts that are the same. In order to distinguish people, especially at the DNA level, you want to be looking at those sections of the DNA that are different from one person to the next. So you have to single out those sections, and for purposes of identifying or telling apart individuals, then you want to look at those sections of the DNA that are not the same from one person to the next.
You described brothers and sisters as being different and yet similar in some respects. Is that right?
Well, they will be similar in some respects because if you think about, you have a mother and a father, and they're going to have children. Each child will not get identically the same characteristic from the mother. So, but if you had 3 children, 2 children might get the mother's hair color, and the third child might get the father's hair color. And the first two children who both got the mother's hair color. One of them might get the father's stature and be big boned and tall, and if the mother was short and small boned, then the other child might get that characteristic, so that even though they had some characteristics in common, they both got mother's hair color, they don't have all their characteristics in common. And that's a very simplistic example, but it works.
So then children would or would not have the same DNA as that child's parents.
The child will not, definitely not have the same DNA as either parent, because the child only gets half of the mother's DNA. So, and the other half comes from the father. So that child is a blend of the mother and father, and the child will not be identical to either the mother or the father.
As far as, and perhaps we can go back to the chart that's currently on the easel, where is DNA found? These chromosomes, do they package DNA molecules or simply a portion of the entire DNA molecule in a cell?
They package the DNA molecules in the cell. One chromosome can generally be considered to be a packaged up DNA molecule.
Okay. Now, this court has heard testimony about serology. Are you familiar with that term?
Yes,
Which, as the jury has heard, involves testing various genetic markers like PGM and EAP. Are you familiar with those terms?
yes.
Is DNA different than the types of testing that go on that test proteins like PGM and EAP?
Yes.
In what way?
The protein markers that are mainly used in serology are coded up. That is, in some, you can think of them as a product of the information that's in the DNA. So when you look at the DNA, you are looking at the most basic level of information that you can look at. When you look at a protein, that is a product of the information that's in the DNA. So you're sort of looking at a secondary level of information.
So if I understand correctly, by typing a protein, you're really looking a little bit indirectly at the particular genetic marker. Is that right?
You are. That's
As a-
right.
As opposed to DNA typing where you're looking directly at basically the blueprint itself.
That's right.
All right. Dr. Cotton, I'd like to take you back and shift topics a little bit and go back to Cellmark Diagnostics itself.
Okay.
First of all, how long have you been with Cellmark?
I've been at Cellmark about 7.5 years. I began to work there in January of 1988.
When was the laboratory itself formed?
The laboratory opened up in October of 1987.
What is Cellmark Diagnostics? What type of work do you do?
We do DNA testing, and we basically do DNA testing for two purposes. One is to answer questions in criminal cases and I'll just give you an example. If you had a blood stain and you had a known individual, could be a suspect or a victim, it doesn't matter, you would attempt to say, could the known individual been the donor of the DNA that was taken from the blood stain? It's exactly the same question that serology is used for in a crime lab. The other type of testing that Cellmark does is paternity testing. And for that purpose, we're usually receiving a blood sample from a mother, from a child, and from an alleged father. And the question is could this alleged father be the father of this child, or is it impossible for this man to be the father of this child? And that kind of testing can be done using DNA.
Going back to your education, and you briefly mentioned the fact that you had a master's as well as a PhD. Could you describe a little bit in more detail when you received those degrees and at what schools?
I received both a Bachelor of Science and a Master of Science degree from Southern Methodist University in Dallas, Texas. And for both of those degrees, I was a biology major. Later on, I moved to California and attended graduate school at UC Irvine and finished a PhD in molecular biology and biochemistry.
You touched a little bit on how DNA played a role in your higher education. Can you describe basically from your undergraduate work through your graduate work, what work did you do in the area of DNA itself?
Along the whole way?
Well, if there's a way of briefly summarizing it, perhaps you can package all three different degrees.
I did, for a Bachelor of Science degree, you take a series of biology courses and you learn about DNA and you learn some biochemistry and you learn some cell biology and you do laboratory experiments. And I don't actually recall that any of my laboratory experiments specifically had anything to do with DNA, but actually it's been a while ago, so I'm not really positive. And for my master's degree, I did do work in a laboratory, but that work involved electron microscopy, which is a method of achieving extreme magnifications, and that didn't have anything to do with DNA. In graduate school at UC Irvine, my research focused on DNA and how it is packaged into chromosomes and how the information in DNA is translated for the cell. And so for the 4 to 5 years that I was in graduate school, um, all of my research work had to do with DNA. And then I took the required graduate classes. And some of those had something to do with DNA and some of them did not.
As far as your graduate work, did it include actual hands-on work with DNA testing itself?
Well, it included a lot of hands-on laboratory work because the whole point of being in graduate school is to learn how to do research. You just asked me whether it had to do with DNA testing. If you're talking about the kind of testing that Cellmark does, no, it didn't because that wasn't being done at that time. It had to do with other aspects of DNA.
All right. As far as your employment, and then once you obtained your degrees, did you then seek employment?
Yes.
And could you describe where you have been employed in the past?
I worked for about 3.5 years in the Department of Biochemistry at the University of Iowa. And I did research there, which was also related to basically to how DNA is packaged up into chromosomes. And then I went to work at the National Institutes of Health in Bethesda, Maryland. And I did research there for about 5 years. And that also involved DNA. And that, at that point now, did involve doing basically the same types of procedures that are currently used in DNA testing.
What are the National Institutes of Health?
It's basically sort of looks like, if you were to go visit there, it looks like a big college campus. And but there isn't very much in the way of teaching done there, or not certainly at the undergraduate level. There's some, but not a lot. And it's groups of researchers, and it's divided into different institutes. I used to know all the various institutes. There's a heart, lung, and blood disease institute. I happen to work in the Institute for Alcoholism and Alcohol Abuse. There's the NCI, National Cancer Institute. So I don't remember how many institutes there are in all, probably more than 20. But these various institutes all together comprise the National Institutes of Health. And the main campus for the National Institutes of Health is in Bethesda, Maryland. And then there are other areas where researchers are located also besides that main campus.
Once you left the National Institutes of Health, is that when you joined Cellmark?
Yes.
In what role when you joined them?
When I joined Cellmark, I was the Director of Research and Development.
Do you currently hold that position at Cellmark or a different position?
A different position.
What's that?
I am now the Laboratory Director.
In that position as Laboratory Director, what are your duties?
My main duty is that I am ultimately responsible for the scientific work that's done at Cellmark. So that's my sort of umbrella overall responsibility. My duties include, along with 2 other PhD staff, supervising the research that's done at Cellmark and reviewing cases that are done at Cellmark. When we receive samples in the lab and we analyze those samples and write a report, the person who has done the work at the bench, which is not me, signs the report, and I and myself or one of the other 2 PhD staff would review the report and sign it also. And I testify in court when required.
All right, we'll return to both of those a little bit later. As far as your position as Laboratory Director, how long have you been in that role?
About 2 years.
This jury has heard testimony previously about an organization known as the American Academy of Forensic Sciences. Are you a member of that group?
Yes, I am.
Are you a member of any other organizations or societies?
I'm a member of the American Society of Cell Biology, the American Society of Human Genetics, and the American Association of Blood Banks.
The American Academy of Forensic Sciences, I assume the title describes it, involves forensic sciences. Is that
Yes.
right? And when you use the term forensic science, what are you referring to?
When I use the term forensic science, I'm generally referring to the work that we would do in the analysis of samples where a criminal case is involved.
The other organizations you described, and I think one of them was, for instance, the American Society of Human Genetics that you're a member of.
Yes.
Is that a forensic organization?
No.
What is it?
It's an organization or a group of scientists that are interested in that is, the scientists that are members of that organization would be interested in some aspect of human genetics. They might be interested in disease diagnosis. They might be interested in gene mapping. They might be interested in forensic science, but it's sort of a generic group of scientists whose overall interest would encompass human genetics.
I think you also mentioned, was it the American Association of Blood Banks that you're a member of?
Yes.
What is that group briefly?
Mostly it's people who are knowledgeable and about the storage of blood and blood typing and the use of blood for medical purposes.
such as transfusions?
such as transfusions, and that certainly that's not my area of expertise. The reason that I'm a member of that organization is that that organization has a program for accreditation of laboratories that do paternity testing, and we are a participant in that program. And they usually at their national meetings will get together scientists who are interested in paternity testing and have a specific forum for that small group. So that's a very small group within the American Association of Blood Banks.
Do you attend meetings as you're able to and as your casework permits of these organizations?
Yes, I do.
Do you present lectures at these meetings?
Not at every meeting, but I do present lectures at some meetings.
On what types of topics?
Generally, it's related to perhaps a new technique that could be used in criminal cases, or a case study that was done in a very unusual case, something like that.
All right. As far as publications, and there is scientific literature in the area of DNA, is that correct?
There's a huge amount of scientific literature generally in the area of DNA.
As far as that literature is concerned, do you attempt to stay current, or do you stay current on that portion of the literature that directly impacts your job at Cellmark Diagnostics?
I mainly am now able to stay current with the scientific literature on DNA as it impacts on human identification issues. I do stay current with some of the other literature, but it's in a relatively narrow area.
You've used this term human identification. What do you mean by that?
Basically, I mean being able to distinguish one human being from another, and as the ability of science gets more and more sophisticated in this area, given enough information, you can distinguish one human being from basically all others.
Incidentally, as far as the literature is concerned, do you act as a peer reviewer for that literature?
I am sometimes asked to review articles that have been submitted to journals and give my opinion on the quality of the work that's in that paper.
All right. I believe you described that one of your duties as laboratory director includes testifying in courts across the United States.
Yes,
Have you previously testified in courts across this country?
yes.
And I'm referring now to the area of human identification as a result of DNA typing.
That's the only area in which I've given testimony.
Could you describe for us approximately how many times that has occurred before today?
I don't keep a running total, but I've probably testified in about 90 or so cases.
Do you have any idea approximately how many states of this country that involves?
Well, again, I don't keep a running total of that, but I'm sure it's at least 20.
Would this be your first time in the county of Los Angeles or not, testifying as an expert in DNA typing?
It's definitely not my first time in the county of Los Angeles.
Dr. Cotton, I'd like to shift gears a little bit and talk with you about DNA typing itself. Is there more than one or just one approach to typing DNA for purposes of human identification?
There's more than one approach.
All right. Are there two primary approaches that are used commonly in forensic science?
Yes.
What are those called, first of all?
The two primary approaches are generally referred to by their an acronym or their initials. One is called RFLP analysis. And that's the acronym standing for restriction, fragment, length, polymorphism. And obviously you don't have an explanation of that yet. The word polymorphism just is referring to differences, meaning it means many forms. So it is a technique that is allowing you to look at many forms of the DNA. And that's one major type of DNA testing that's done in this country today, and actually many other countries as well. The other type is referred to as PCR. Now, there are many different kinds of PCR tests, and PCR is the basic procedure that allows you to do the test. And PCR, the initials stand, and again, this won't make any sense yet, the initials stand for the polymerase chain reaction. It's a procedure, and that procedure can be applied to different types of genetic markers and different types of tests. But any test that uses that procedure is generally referred to as a PCR type test.
I'd like to start with the RFLP process first, if we could. And first of all, could you describe for this jury, taking weeks, what the RFLP process is and how it actually is conducted?
You mean, could I take several weeks to describe it?
Yes.
Oh, I'm sure we could.
To
I
your,
don't think we want to do that.
to your knowledge, are we going to in fact limit it to something substantially shorter than that?
We're going to, based on my discussions with you, we're going to try to make it much shorter than that and pick out just those sections that are really important to have an understanding of what the final result looks like and what it means.
Okay. This RFLP process, when was it developed?
It wasn't developed on a special day. Um, it's a series of procedures that all tied together are called RFLP. Some of those procedures were developed in the mid 70s, and some of the information that scientists needed to actually finally put all this together was not developed until the early 80s. So you can't give it a date, but you can give it a time frame from about 1975 to about 1985. The information that's used in RFLP testing for identification was discovered during that time frame.
Was there a time when locations on the DNA molecule that you've already described were found to be different amongst people, as you earlier testified to?
That's been known for a long time. Early on, of course, it was just known sort of intuitively that there had to be sections that were different since people are very different. The information about heredity and how it relates to DNA was described back in the late 1800s and early 1900s. But the discovery of the specific types of differences that this test looks at was basically about 1983, 84, and 85.
Was that as the result of, in particular, one or two scientists in this country and outside this country?
Yes.
Who were those individuals?
The two major contributors in this area, and again, it's very hard to say this because without previous information, they wouldn't have been able to make their major contributions. So you have a whole series of scientists that are kind of contributing here. But the two contributors to the kinds of information we are looking at in this testing would be Dr. Ray White from Utah and Dr. Alec Jeffries from the University of Leicester in England.
Incidentally, was Dr. Jeffries in England honored for any of his work in this area?
Yes, he's received several honors, including being knighted for his discoveries in this area.
By knighted, does that mean he's addressed as Sir Alec Jeffries?
I believe I know it's Sir, and then he's also a professor and some other things. So there's sort of a series of Sir, Professor things that go in front of his name, but I don't know that I could get it right.
I think they're called titles.
Did Dr. Jeffries play a role in any manner in the formation of Cellmark Diagnostics?
Basically, he did. The University of Leicester, where Dr. Jeffries works, owns the patents to his discoveries. So the series of events was he made these discoveries, certain of this information was patented, and the parent company that owns Cellmark Diagnostics purchased the rights to those patents, and they, along with Dr. Jeffries' help, did the developmental work to take it from, excuse me, being a procedure that he did daily in his lab to having a protocol that could be followed in what's more like a production lab where you're doing the same type of test on each sample that comes in the lab.
Is there any relationship between your laboratory in this country as well as Cellmark in the United Kingdom?
Yes.
What's that relationship?
We are both owned by a parent company. The name of that company is Zeneca.
Was Cellmark in the United States formed before or after Cellmark in the United Kingdom?
I think it was just about a half a year after the formation of Cellmark in the United Kingdom.
As far as the procedures that were used at Cellmark in this country at its formation, did they have anything to do with what had been used before or even at that time in England?
At the time that Cellmark began doing casework in the United States, we were using the identical procedures to those that were used in England. Now our procedures are probably not identical. They don't do very much testing for criminal cases in that lab anymore. And so our procedures would no longer be identical, but they would still be very similar.
Incidentally, why don't they do as many criminal cases now in England?
Oral.
There is a very large lab in London, and they do almost all of the forensic testing for the United Kingdom.
Now, with regard to, and you've used this term RFLP typing process that we'll get into in a little more detail later, is this RFLP testing approach used exclusively in human identification?
No.
What else is it used for?
Let me give you an answer in sort of two phases. A very similar procedure can be used to identify animals. And in fact, There is a lab in Oregon and another lab in Canada that uses the same types of procedures to look at poaching. If somebody has some meat in their freezer and they say, well, this is just a deer, and the I don't know what the organization would be that would be checking on this, but whatever organization it is, they say, well, no, this is some endangered pronghorn antelope, or I'm sort of making this up, as you can see. Then they can use these same kinds of procedures to determine whether or not the meat in the person's freezer is in fact from a deer or from the endangered species. So that's a very analogous type of thing to what's being done in a criminal case. The general procedures that are used in this testing are used in genetic diagnosis and in gene mapping and in a lot of other research applications other than human identification.
Let me stop you and ask you about when you use the term genetic diagnosis.
You have a donor and you have a recipient of the bone marrow from the donor. And after the transplant is done, you can use these procedures to follow whether or not the transplant has been effective by looking at the blood cells in the recipient and saying, is the original genetic source of those cells those of the recipient, which you're hoping it won't be, or those of the blood marrow donor, and you're hoping that it will be that. And you can track the progress of the transplant using the very same procedures that are used for paternity or identification testing.
As far as disease diagnosis, if I can return to that, is this RFLP procedure then used to determine, for instance, whether an individual suffers from cystic fibrosis?
It can be. There are now easier tests than the RFLP procedure, but that was certainly early on the procedure in the research stages that would have been used.
You also brought up the use of this technique, for instance, in the poaching scenario. Is this RFLP technique also used to help save endangered animals?
Yes.
What about this technique, this RFLP technique, in, for instance, plants? Does it have any role there?
There are the main role that I'm aware of, and I'm not a plant biologist, so there may be many other aspects that it's useful for that I'm not aware of, but in plant breeding, where a company has a corn hybrid that has specific characteristics, those characteristics can be identified and tracked in that hybrid using RFLP analysis with exactly the same types of procedures that are used in testing for paternity.
Does this RFLP testing technique play any role in, for instance, identifying the remains of American war dead?
It has, yes.
And does that include, for instance, remains from soldiers killed in battle?
Yes.
Now, you've described a number of general areas, including human identification, that this RFLP technique is used. Would the human identification use of this technique be the majority or most or a small portion or what?
Overall, it would be a small portion. If you look at anyone in science who might use this procedure or this set of procedures that makes up RFLP analysis, human identification would be a very, very small fraction of its total application.
Is this a technique that was basically born out of forensic science, or did forensic science borrow it from the greater scientific community?
It would be that forensic science borrowed it from the research community.
What about in terms of how wide this technique is used? And I'm talking about geographically. Is it used in the United States and England alone, or is it used elsewhere?
Sustain the phrase question.
As far as the RFLP typing approach, is it used strictly in English-speaking countries, for instance, or is it used in some wider area than that?
No, it's used in a much wider area than that.
And would that include, for instance, in countries around the world?
Yes.
Now, if I can return to the RFLP method, first of all, would it assist if I asked you to describe briefly this method to use, again, the drawing pad for purposes of demonstrating what happens?
I probably can't do it without scribbling on the pad.
Okay, fair enough. Marked as People's 241.
241. Call this RFLP drawing. Mr. Clark.
Thank you. In fact, Dr. Cotton, if we could, before we do that, could we go back and perhaps take a step down? I'm going to ask you to put just very brief titles on the previous exhibits, the previous drawings. Perhaps we could start with the 46 chromosomes chart that I believe is People's Exhibit 236. No, 235, excuse me. That's labeled 46 chromosomes. Would that be an appropriate title for that chart?
It will do.
Okay, perhaps you could just underline it, and then we'll underline the titles on each one. And
All right,
then-
235 will be labeled in chromosomes, underline, 46 chromosomes, underline.
all right, and if you could go to the next drawing that you made, which is exhibit 236. Does that drawing depict on the left the single pair of chromosomes and then the two strands of DNA to the right?
Yes.
What would be a short title for that drawing?
Why don't we just put chromosome pair?
Okay.
Does it matter where?
Why don't you put it in, if you have a different color, I think you could put it in the middle, it'd be fine, as long as you underline it. And then as far as the two strands of DNA to the right, would it be appropriate to give that a title?
No, that's just two strands
All
from
right.
these two chromosomes, so that's still a chromosome.
Okay, if you would go to the next diagram, which I think has already been titled double-stranded, would that be an appropriate title for what I believe to be Peoples 238?
Sure.
Okay, and that just leaves us then the next page.
Let's add DNA.
Okay, and then if you would go to 239, the last drawing. And is that a demonstration of single-strand DNA?
Yes.
Does your T have a cross on it? And again, Your Honor, that would be People's Exhibit 239. Now, Dr. Cotton, let's provide you with a new page. Which will be People's 241, I believe. First of all, why don't we label it at the top, RFLP method? And if you would now use that particular piece of paper to help describe this RFLP testing process.
Okay.
Go ahead.
No, you just- What I want to do is go back to the idea that you have two chromosomes, or you have a chromosome pair, and that we're going to look at the DNA from that specific chromosome pair. And I want to explain to you we keep talking about looking at differences in the DNA. I want to show you an example of the kind of difference that we're looking at, because I think that's the only way it will really make any sense. And then, once we've done that, we can sort of walk through the procedure.
All right.
So let's go back and say we have our two chromosomes worth of DNA here. And they have, the A and T and GC base pairs all along. And so if we're looking around on these chromosomes, you will come to a section that contains what is referred to as repeated sequences. And I'm going to make a very short and simple, short meaning short in physical length, simple example of this, but this is not an unrealistic example. Suppose we come along and we're looking down the sequences of bases, then we see that we have a C followed by an A followed by a T, with another C followed by another A followed by another T, and another C followed by another A followed by another T. Now, what I'm going to do is only draw one side, so if you'll just assume with me that the other side has the appropriate matching base. So we have this sequence, CAT, repeated 3 times. Now, if we went and looked at the other chromosome, this is now, from one person, and you're going down the chromosome and you're looking at the base pairs, and you come to sort of a matching section, and you find that it has a C followed by an A followed by a T. And you see that that sequence of 3 base pairs is repeated quite a number of times. But on this chromosome, it's repeated more times. Here we have, on the one on the left, we had 3 repeats. And here we have, on the one on the right, we have 5 repeats. So we have this short section of something that's repeated over and over again. Now, you have to sort of think about if we made this same drawing for yet another individual, and another, and another. What you would find is that another individual might have a section, they'd have this, you go on the chromosome, they'd have this same repeat, CAT. But it might be repeated 10 times on one of their chromosomes and 18 times on the other. So that everybody has the CAT sequence on their chromosome, but not everybody has the same number of repeats. That is the difference that RFLP testing looks at. So if you imagine in your head that we had a method to cut out this section, and then you add to that that we had a method to measure how long this section was that we cut out. For this person, if we could do that measurement, we'd have a section that was 9 base pairs long. And for their other chromosome, we'd have a section that was 15 base pairs long. So this one becomes 9, and I'm going to use the abbreviation BP to stand for base pairs being the GC or the AT pairs. And over here, our 5 repeats is equal to 15 base pairs. So, and then if we had another person, and we could make another diagram just like this, if they had a chromosome that had 10 repeats, that would be a section that was 30 base pairs. So it's those lengths that are different from person to person. And in this abbreviation, the P standing for polymorphism, the L stands for length. This test looks at length differences in the DNA, and the length differences are created by the number of repeats that happen to be end to end at a specific location on a chromosome pair.
So is it then the case that this RFLP method is simply a tool to determine how we vary from one another as human beings at this particular region of DNA where these repeats vary?
Yes.
As far as the method itself, to your knowledge, do we have a prepared chart that illustrates how this difference is actually determined using the RFLP test?
We have, you have, and I've looked at a prepared chart that doesn't illustrate this, but illustrates the set of procedures that allows you to ultimately look at these length differences.
And is it these length differences that are simply what you're looking for as a DNA scientist when you're using this RFLP method for identification?
That's right.
All right. And Your Honor, with the court's permission, I'd ask be marked as, I believe, People's Exhibit 242,
Equals 242.
a fairly large prepared diagram.
12 hours isn't really that long for labor. It's a lot of women are way more. My first one was 24 hours. Anthony was 12 hours. My middle one was different. Like 12 hours is not that long. It just takes a while. Takes a lot of contractions.
Entitled, I believe, the RFLP method.
Let's see what Google says is the average time. So 12 hours is the low bar. It's the low. But that's just for your first kid. The other kids go a little faster. Early labor is 8 to 12 hours. Active labor, 3 to 5 hours. Whew, yeah, I remember those. Hoo, hoo, hoo.
Now, Dr. Cotton, first of all, as you've described on your drawing, People's Exhibit 241, where you've described this differences amongst us as human beings in the number of times we repeat a particular sequence, how are these particular regions of DNA picked out or selected to use for identification purposes? Does that question make sense?
It makes perfect sense, but it takes that whole procedure to do that.
Okay, all right, then why don't we go ahead and, if you would, then describe this RFLP method in the basic steps that are delineated, and perhaps I can ask a question first. Have you had an opportunity to look at this large chart entitled the RFLP method that was previously prepared before coming to court today.
Yes, I have.
And in that review, does this diagram basically describe the individual steps that are part of this RFLP DNA typing
Clark,
method?
excuse me, Clark, let me interrupt you just one. Let me see counsel left court.
Wait, what? How in the world do we have a sidebar? What is so controversial that the judge needs to pull the lawyers aside right now? I think I blanked out. I think the loudest I've ever snored in my life was after my first one. The poor nurses had to hear that. I wonder what they're talking about. DNA?
Thank you, counsel.
All right, Dr. Cotton, with regard to this diagram, then, could you describe would utilize this diagram and the drawing you previously made to describe how this RFLP typing method actually works?
The diagram on the board has everything numbered, so I'll try to relate what I'm saying to number. So let's say you have a sample, some biological sample. The diagram illustrates either a tube of blood or blood stain, but this could be, as I said before, a hair root. It could be a semen stain. We'll just say we have some biological sample. And the first thing that happens is that the DNA is extracted from this sample. What that means is the DNA is purified from the other components of the sample that you don't care about. The DNA is in the cell and you have all these proteins. Proteins are nice, but for purposes of DNA testing, we don't need them. So you're purifying the DNA away from the other cellular components, which include proteins and cell membranes and things like that. That's step number one.
All right.
Step number two on the chart says the DNA is cut into fragments by a restriction enzyme. When I said a little while ago that if you had a method of cutting out these sections where the repeat exists, that you could look at how long it was. The method is to use a protein that is called a restriction enzyme. And these enzymes have the characteristic that they can locate a specific short set of base pairs. Experimentally, it's predetermined there are many restriction enzymes. There are over 100 different restriction enzymes. And let me just give you an example so it doesn't sound so briefly. Let's just say you have a double-stranded piece of DNA and you have this series of bases C, C, G, G, and you go down the DNA and you find another set where it happens to have C, C, G, G. If you have a restriction enzyme that recognizes this site, it would make a cut wherever it happened to see a CCGG. And I'm just giving you one example. There are many others. So
Actually, while you're on that drawing, Dr. Cotton, perhaps you can label that restriction enzyme.
well,
Would that be an appropriate description or not?
let's call it this is just an example of a restriction enzyme cutting site.
Your Honor, could that be marked equals 243?
243.
Thank you. All right, continue if you would, Dr. Cotton.
I'm going to go back to the previous diagram. The test is set up so that you are using a specific restriction enzyme that we know will cut outside the area of the repeats. So the enzyme that's used in the test is designed so it will cut at the beginning, just before the beginning of the repeat, and just after the repeat, or somewhere close to that. So you purify your DNA, you add your restriction enzyme, and you make these cuts. Now the enzyme cuts many other places. So basically, if you go back to the spool of thread analogy, and you've rolled out your spool of thread, and it's very long, you've now taken a pair of scissors and cut it up into a whole bunch of small pieces. And among those small pieces are these two that we're interested in.
All right. What step do you take next to locate those interested fragments?
The next step, actually, the next series of steps are the steps that allow you to locate those fragments. So the first thing you do, now that you've got all these little fragments, is that you use a procedure that's called electrophoresis. It's used, it has many other uses. You can do electrophoresis with proteins, but in this case, we're doing electrophoresis with DNA. And that electrophoresis will allow you to separate out the pieces according to how big they are. So if I had a test tube full of pieces of DNA that were all different size, at the end of the electrophoresis, it's as if I had been able to reach in and put the small pieces down here and the medium-sized pieces over here and the larger pieces down at that end.
If this jury had heard previous testimony about the use of electrophoresis to type proteins such as PGM and EAP, would this be the same tool that you're talking about in the RFLP typing process?
It's very similar, but it's not exactly the same.
All right, go ahead.
The picture on the diagram that's numbered 3 is the illustration of separation of the DNA fragments on an agarose gel. That illustration is pretty okay, but let me try my own and see if I can make it a little more graphic. The gel is about the size of a little smaller than an 8 by 10 piece of paper. Actually, it's about the size of your notebooks, a little bit wider and about that long. It's about 1/4 of an inch thick, and it's made of agarose. Agarose comes as a powder, and you boil it up sort of like Jell-O, and it goes into solution, and when you pour it into a tray, it solidifies. So it has some of the same characteristics of gelatin, although it is not gelatin. So you're pouring this gel into a tray, and if I look at that tray from the side, after I've poured the gel, this is about 1/4 inch thick here. I also use a small mold to make an indentation at one end. If I take that same gel and I draw it as if it was sitting on the countertop where I could look down on it, I would have a series of these rectangular indentations across the top of the gel. Now on the diagram, they're sort of showing you where 2 samples have gone in that gel, so the red lines, the red lanes are sort of one that represents one indentation where you could put a sample. Anyway, you have your DNA sample, and the sample is placed into this indentation in the gel, which is called a sample well. And the DNA moves through the gel. The DNA has a charge. It carries a slight negative charge, so if there's a positive electrode at this end and a negative electrode at this end, DNA is forced to move through the gel, and the gel acts like a sieve. Short segments of DNA can move through the gel rapidly. They can work their way through this sieve pushed along by the electric current. And shorter pieces will move through the gel the farthest. The longer strands of DNA cannot move as rapidly through the gel, and they do not go as far. So this is the way of taking these 2 pieces of DNA. These are now the pieces that we're interested in. Remember, they're part of the group that we're looking at. We have these 2 pieces in our mass of bits. We want to see where they are. This piece is shorter than this piece. So in the gel, the shorter piece, let's say, moves this far, and the other piece cannot move as far. So now these 2 pieces are separated. The piece that was only 9 base pairs is in a different position than the piece that was 15 base pairs.
All right, could we, with this diagram that you've just drawn, would electrophoresis be an appropriate title?
Sure.
And I believe that would be People's 244, Your Honor.
All right, Mr. Clark, I need to allow the jurors to take a comfort break at this point. Ladies and gentlemen, I forgot to mention to you this morning, I don't know if the bailiffs mentioned to you that we have a modified court session today. I have a funeral to attend this afternoon, so we're going to go through until 1 o'clock and then break at 1 o'clock. So we'll take a 10-minute comfort break at this point, and we'll resume again at 12 sharp. All right, please remember my admonitions to you.
of all, and I'd like to direct you specifically to what's labeled number 3, low gel, and the photograph above that. Does that appear to be a gel?
Yes.
All right. Is that the same, different, or similar from the gels that you're describing as part of the RFLP process?
Well, I'm sure it's different. I'm not exactly sure what kind of gel that is. If it was the RFLP process, and that looks to me like a glass plate, although I can't tell that well. If it was the RFLP process, it would be a plastic tray that has sort of like pouring something into a square or rectangular cake pan, but the edges of the tray are maybe about 3/4 of an inch high.
Okay. Then taking you on to step 4 on this diagram, People's Exhibit 215, it's labeled 4, electrophoresis. Is that the same or similar to the electrophoresis process you've described in the RFLP method?
The tank that's shown in photograph 4 doesn't look very different from the type of tank we use. It has space at either end where buffer or some kind of a wick goes, has a plastic lid that comes down to protect the user. What's really different here is that if you look at photograph number 3, where it says low gel, first of all, the position where the samples are loaded on an RFLP gel is very close to the top, more like what I've drawn in this diagram. Whereas there, it's maybe 1/4 of a way down the gel. And these are, in the RFLP procedure, these are liquid samples that are loaded using a pipetter with a disposable plastic tip.
Okay. And then lastly, on this Exhibit 215, where it's labeled running the gel, do you in fact run gels during the RFLP method typing process?
Yes, it shows there a series of gels and 2 power supplies up on a shelf, and we have a similar looking power supply. It's connected to the gel tank such that you have a negative and a positive electrode, which is creating the current, the electric current, which is pushing these DNA fragments along through the gel.
All right. Your Honor, with the court's permission, could 215 just be brought over towards this side so the remaining jury members could have a view of it?
Yes.
Now, Dr. Cotton, at the time the electrophoresis, or step 3 on People's Exhibit 242 is completed, can you see these different fragments on the gel?
No, you can't see.
What's the next step in the process?
The next step in the process is to create a permanent record of the separation of the DNA fragments in the gel. If you tried to pick up an agarose gel with your hands, it would be like trying to pick up a 1/4 inch slab of Jello. It would break apart in your fingers. So whereas all the information you want is in this gel, you have to have a permanent way of storing this information. Can I backtrack for 1 second
Sure.
and then start a new diagram?
Yes.
Let's still consider this 2 chromosome or 2 DNA pieces from the diagram a few places back. Let me make sure that I straighten one thing out and then go on. I talked about a fragment of DNA that was 9 base pairs and another fragment that was 15. Let's put this more into reality for this test. The sizes we might look at, you could look at a size that was 900 base pairs and a size that was 1500 base pairs. This is more like the size range, and actually you can look considerably larger than this, but this is more like the size range of pieces that you would be looking at in this test. Still, due where the differences in size were due to a different number of repeats in that section of DNA. So that if we go back, we have our 900 base pair piece has moved further down through the gel, and the 1500 base pair piece has not moved as far. When you have this separation in the gel, this is the information that you really want. And the entire rest of the procedure are the set of steps that allow you to visualize where these pieces of DNA went. So if you understand that where the DNA came to in the gel is a measure, an indirect measure of its size, that is, this distance traveled is an indirect measure of the size. The further the distance traveled, the smaller the size piece of DNA. The shorter the distance traveled, the larger the size is. This is the information you're after, and the entire rest of the procedure is how you see this visually.
All right, Your Honor, for the record, the witness has been referring and writing further on People's Exhibit 244.
Yes.
All right, Dr. Cotton, then would you be, or are we proceeding to step 4 on the diagram, the RFLP method?
Yes,
And could you describe that, please?
yes, and I won't, actually, maybe I won't draw another one because this is pretty nice here on step 4. What you want to do now is keep this record, or make a permanent record of where these DNA fragments are. And the way that's done is to transfer the DNA in the same relative positions from the gel onto a nylon membrane. And that step, or that process, is illustrated in step 4.
All right, would a laser light pointer help you? The diagram or not?
How about the good old fashioned pointer?
Or a pointer, if you can reach. I think we've got a pointer right next to the entrance to the. In fact, Dr. Cotton, did you bring one of these membranes to court today?
I gave it to you, and presumably you brought it to court.
At this point, I hope I did. All right, Your Honor. I'd ask to have marked as People's next in order, I believe, 245.
44, I believe. I think it is 244. Mrs. Robertson, 244? 245, I stand corrected.
And I have provided the defense one of these items as well.
All right, proceed.
Dr. Cotton, if I can show you what seems to be a object about the size of what, 8 inches by 8 inches or so?
Yes, it's actually 20 centimeters by 20 centimeters.
Okay, that sounds like a more precise description. What is, and referring you to what will be marked People's Exhibit 245, what is that?
This is how the membrane comes to us. It's in these red pieces of, this is just a piece of paper. And it has written on it MSI, and that's the manufacturer of the membrane that we use. And sandwiched in between the two pieces of paper is a nylon membrane. Now, if I were doing this in the lab, I wouldn't be touching it with my fingers. You would be handling it with gloves because fingerprints wouldn't be helpful. Any of the oil from your fingers is really a problem. And this hasn't been used in the lab, so it's clean. And this is the nylon membrane. So in the example here, you have a sponge, and in our lab we use something that's called a wick, but it's all going to do the same thing. So even though this is slightly different from my lab, I'm just going to stick with this. And then on top of this, and the sponge is soaked with some solution, and on top of the sponge you place the gel. So you can see the gel here is green, and you have a green layer here. That's the gel. Of course, the gel isn't really green; it's really kind of just white. And you can see that the membrane isn't really orange, but they made it orange. And anyway, so you have that gel, and you simply, if I had a gel here, I'll use the top of this thing. You would very carefully take this nylon membrane and lay it down.
Actually, I'm sorry, Dr.
Gel.
Cotton, perhaps you could demonstrate that on the actual bar in front of the jury so that more can see.
I had my gel sitting right here. I would carefully lay this nylon membrane down on top of the gel and let it fall down easily. And it would be wet beforehand, and the gel is damp. And then you can, when I did this in research, I used my fingers, but in the lab we now use a pipette. But you would smooth down the nylon membrane so that it was in easy, close contact with the gel.
All right, Dr. Cotton, could you keep your voice up, please?
Yes, sir, thank you. Now, what's the purpose of this is to have the DNA move out of the gel and come in contact with the nylon membrane. If you go back to the diagram here, the salt solution that's in the sponge will move up through, oh, well, wait, let me finish the order here. We have the sponge, the gel, the membrane, a big stack of paper towels, and a weight. So the idea is that the solution moves up, that's soaking in the sponge, moves up through the gel and carries with it the DNA. And it's sucked up, the solution is sucked up into the paper towels, but the DNA becomes trapped on the nylon membrane. So if you were to go back to this diagram, the membrane would be layered right on top of the gel, and the DNA would move up through the gel and become attached to the nylon membrane. The paper towels and the weights serve the obvious purpose, the same as if you had a puddle on the counter and you put the paper towels on it, the solution would be soaked up into the paper towels. It's not a very elegant contraction, but this is one of the most important procedures in all of molecular biology, and it's used all over the world.
Mr. Clark, the diagram that Dr. Cotton referred to, again, the drawing, I believe that's the electrophoresis diagram.
Correct, 244.
Now, I have omitted in one important feature here, and somehow I always have trouble getting this in the right order. Just before you set this setup up, you treat your gel so the DNA fragments that are double-stranded will come apart and become single-stranded. And you do this by soaking the gel in a solution of sodium hydroxide. So basically, the other method that we talked about was heating it, but if you heat it, the gel will just melt, so you can't do that. So you're going to separate this, what was double-stranded to now be single-stranded in the gel, and then you're going to do this transfer so that as the DNA is bound to that nylon, it's in a single-stranded form. And that's the important thing that I left out.
Actually, two questions if I can. First of all, why doesn't the DNA travel through the membrane and up into the paper towels?
DNA binds to the membrane and it becomes trapped there. The membrane doesn't have sufficiently large pores in it for the DNA to get through.
Okay, and then you also referred to this process as not being particularly elegant, is that right?
Well, it's very elegant, but it's not very fancy. It doesn't require a lot of equipment. You don't need any electricity. You could set it up in your kitchen if you really wanted to.
Is this particular step limited to forensic science, or is it applicable, is it actually used in any technique involving use of the RFLP method?
It's used in any technique where RFLP is used, and it has other uses as well. But if you go into any of the scientific journals, you probably can't go through a journal that talks about molecular biology or biochemistry without finding an article where this procedure is used. This procedure is called a southern blot, and it doesn't have that title. Oh, yes, it does have the title here on the diagram where it says by a technique known as southern blotting. It's called a southern blot because the scientist who devised this procedure in the late 1970s, mid 1970s, his name is Ed Southern, and he's from England, and his last name became attached to the procedure because it's so widely used. So it's called a southern blot. That's the name of the scientist who devised the procedure, and it's been used since that time.
All right. So now is it correct then that this membrane that's really marked People's Exhibit 245 in this process now has single-stranded DNA attached to it?
Yes, and the important thing is, however the strands were separated in the gel, they are now, that is, however the DNA fragments, that is the large one versus the small one, were separated in the gel, that same spatial separation is now on the nylon membrane. So the DNA is attached to the same nylon membrane, and the pieces are in the same relative positions that they were in the gel when the gel was turned off.
What's the next step in this RFLP typing process after you have the membrane with the DNA attached to it?
The next step, I have to read what's on the board for a
All
second
right.
to see where I am on this. Okay, so over here, after the blot, you have this statement on the board, which is correct. The DNA fragments are now on the nylon membrane, but are not yet visible. Now, actually, it shows you as if something is visible, but really, when you put the nylon down and you take the nylon off the blot, it looks just plain, just like you saw it. It doesn't, except it's wet. And now you get to the point of the next few steps that allow visualization. And here's where it becomes important that you understand that you can take DNA fragments apart and put them back together, because now you're going to take what's called a DNA probe, which is simply a piece of DNA that you've made in the laboratory and has a radioactive tag on it. It's single-stranded. And if your repeat was CAT, your probe would be GTA.
Actually, you used an earlier drawing that demonstrated a tag, did you not?
I did.
Perhaps you can refer back to that. And Your Honor, that would be People's Exhibit 239, labeled single-stranded DNA.
So in my single-stranded DNA drawing, I didn't draw the repeat CAT, but let's suppose this is our repeat. if this is our repeat, the DNA that's on the nylon is now single-stranded. So you have this available to collect the second strand, and you would have the other side available to collect a second strand. So you take the nylon membrane and put it in a plastic tub, or you can put it in a tube-like thing, but I'm going to use the example for a tub because it's a little easier. We used in the lab a Tupperware container, a square one, in which it was just about the size of that nylon membrane. So the membrane would be flat in the bottom, and then the membrane is covered with a salt solution. The radioactive DNA probe is added to that solution, and that solution is let sit at a specific temperature overnight. So overnight being, about 16 hours perhaps. And during this time, this radioactive probe DNA, which is simply, the probe is actually, for what we do in the lab, attached to C's and G's. During this time, this probe that you've added will zip up with any DNA on the membrane that it can appropriately zip up with. The radioactivity is something that you can follow. If you take your membrane, add your probe, let it go overnight, take your membrane out, rinse off the excess probe. If I held a Geiger counter up to it, I would get a few clicks, a Geiger counter being able to detect the radioactivity. But that's not accurate enough. So instead of a Geiger counter, you would then, if we can go back here, here's our membrane that now has some probe bound to it. To detect where that probe has bound, you lay an X-ray film over the membrane. You do this in the dark. You put it in a metal cassette that sort of looks like two book covers that then close up together. And the radioactivity that's on the membrane will expose the silver grains on the X-ray film, creating a dark spot or a line. It's really a line. And that is the image of where the probe bound to the DNA. So if we go way back, we go back to this diagram. This was originally our gel. Now this pattern of DNA is on the membrane. And now you've added a probe to it, and the probe is bound specifically. We make the probe so it binds specifically to the CAT repeat. That's what it's going to do. So the probe is bound specifically here and here. And it's been washed off everywhere else because only in these two positions is there a piece of DNA that it can come back together with. And we've laid a piece of X-ray film over this membrane and allowed it to expose. And on the X-ray film, a dark line will appear in these two relative positions. That dark line tells you where the DNA fragment went to in the gel when the gel separation of these pieces occurred.
All right, referring to People's Exhibit 244, Your Honor, the drawing.
Yes.
Dr. Cotton, these probes then, are they specifically designed to look for that repeat sequence that you're searching for on the membrane itself?
Yes.
And when they recognize the sequence that they're looking for, the probe then actually mates up or zips together with the DNA that's already on the membrane from this blotting procedure?
Yes.
Is the probe then simply a means for you to be able to see where the fragment is on the gel that you're actually looking for?
Yes. The probe, this is your tracer, it's your eyes, it's your tag. And we talk about it like it was a thinking thing, but of course it's just a piece of DNA, and this is just a chemical reaction that allows these two DNA strands that were single-stranded to come together. So it's simply your way in the laboratory of identifying where these pieces of DNA went that had the repeat that matches to the probe.
Then is the use of this radioactivity and X-ray film so that you can actually look and see where these fragments are?
Yes, that's exactly what you're doing.
And is that done by your own eyes?
When you look at the X-ray film, yes, you're looking at it.
Does the X-ray film have a particular name in the RFLP testing process?
It's properly called an autoradiograph. In the lab, we refer to it as an autorad. It's just simpler to say, and that's what most people would refer. That's the term most people would use.
All right, Your Honor, at this point, I'd like to have marked as people's next in order.
Is that 246? All right, People's 246, Mr. Clark.
Yes, a single, what I believe to be an autoradiograph. A copy which has been previously provided to counsel.
Right, single auto read.
Is that what they're doing? I have no idea. I have no idea what's going on. None.
Dr. Cotton, just showing you what will be marked People's Exhibit, I'm sorry, 246, right?
246.
What is that?
This is a copy of one of the autoradiographs that was produced in this case.
And is that typically what an X-ray or autoradiograph looks like following the use of this RFLP typing process?
Yes.
All right, without getting into what the samples are on that particular test, with the court's permission, could Dr. Cotton simply hold that up so the jury can see it from a
Yes.
distance?
This is one of the film. Well, this is a copy, but the copies are very good. They don't. So if I
Sustain. Mr.
All right.
Clerk.
All right, Dr. Cotton, if you would just hold it up at the moment, and we will return to this particular autoradiograph later. But is this typical of what such an X-ray looks like at the end of this RFLP procedure?
yes, it is.
Okay, and is
Without
that
all the lettering,
okay?
you have to subtract out the lettering here and the lettering down here.
The lettering you're referring to at both the top and bottom of this particular exhibit, is that done to aid in knowing which sample is which, basically?
Yes.
Okay. All right, fine. Would that autoradiograph then represent what is labeled on the RFLP method chart at step 7 and then step 8?
It would only represent what's labeled in step 8.
Okay. So step 7 is describing the creation of this X-ray film in its early stages by laying it on top of the membrane.
Yes.
All right, very good. Perhaps I can take that back. And if you would, could you have a seat back on the witness stand?
And Mr. Fairhill, can you rescue the diagram easel there, please?
Actually, with respect to the RFLP method, the large chart, could that also be brought down so that all of the jurors could have a look at it?
Fairhill, Mr. Clark, perhaps you should both try to handle that. Yes. All right, thank you, counsel.
Like questions like, why are you asking so many stupid questions?
Dr. Cotton,
That
is
would be a
that then
good
a
question
capsule summary of this
from the
RFLP
jury.
typing approach?
It is.
You are familiar with the term controls.
Yes.
What is a control?
Control is usually some kind of sample that helps you to determine whether all the procedures that you just finished using worked as they should. And it usually is a sample that you know what the results from the control should look like. And if they look appropriately, then it gives you some information that your test was done appropriately.
Now, referring to this RFLP typing process, do you utilize controls as part of that testing?
Yes, we do.
Are there a number of different types of controls?
Yes.
How do you know when a control isn't working properly?
Well, it depends on what kind of control you're using, but for this procedure, you are ultimately looking at that X-ray film and visualizing your result, and later on you might make some measurements. But if we just talk about looking at it for the time being, the controls have a very typical appearance, one that you are accustomed to seeing. So if they look as they should, then you would make a general first impression assessment that the test and the procedures that you used had been done appropriately.
What if the controls didn't work properly in a particular test?
Generally, if they haven't worked properly, you would either go back and repeat it if there was a possibility to do that, or if you couldn't repeat it, you would probably just say that your results are inconclusive, because if your controls haven't worked, that there's some implication there that how you worked on the actual samples may not have worked either.
Now, this autorad in People's Exhibit 246 represents an autorad from an RFLP test. Is that right?
It does.
This autoradiograph, is it in fact preservable? In other words, is it in a form that, for instance, can be looked at a later time or by other people?
Yes, the autoradiograph is your permanent record of the results of the test.
As far as the RFLP typing results, and first of all, did your laboratory obtain RFLP typing results in this case?
Yes, we did.
Are those RFLP testing results reflected on autoradiographs, as an example of which is People's Exhibit 246?
Yes, they are.
Now, I'd like to discuss a different area with you, and it relates in particular to when you are reading results from an autoradiograph. First of all, do you do that?
Yes.
And you look for certain things, is that right?
Yes.
If I use the term match, what does that mean to you?
It means to me that on the autoradiograph, the position of two DNA bands, or possibly the positions of all the bands from one sample to another, are so close as to be visually that I couldn't distinguish one from the other.
What does that mean, that you can't distinguish one from the other?
They look alike,
Does that have a particular meaning in this RFLP typing process?
of course.
What's that?
From person to person, there's so much variation in the length of the fragments that have a repeat that if you look at several genetic locations, and for each genetic location, two samples are the same, and you look at another and they're the same there, and you look at another and they're the same there, and you keep doing that, the more genetic locations at which two samples are the same, the higher the likelihood that the two samples are in fact from the same person.
All right, Your Honor, if I may, return to People's Exhibit 242, the large board, because I believe it has an item that I'd like to ask the witness another question about. Put it away too quickly. Thank you. Dr. Cotton, referring you to what would be step 8 on this board, People's Exhibit 242, does that particular depiction, would that help you in describing what you mean by a match?
Yes.
All right, could you then go ahead and describe that? And if the pointer would help, please feel free.
This diagram and the things that I drew on the chart are illustrating the kinds of results you would get when you looked at one location on one chromosome. So since you get two copies of the chromosome, you get one piece of information that you inherited from your mother and the other from your father. And so if we look at this sample that would be basically on your left, you have two dark lines on the autorad indicating the two DNA bands that are from this sample. And then if you go on to the middle sample, you have two bands on this autorad from this middle sample. And then if you go to the third sample, you also have two bands. The middle sample and the sample on the left that I'm pointing to, the bands are in essentially the same position. So it is possible that sample number 1 and this middle sample, sample number 2, could be from the same person because they have DNA bands that are in the same position. This third sample on the right, which also has two DNA bands, these bands are clearly in a different position than either the first or the second sample. That tells you immediately that the DNA in this third sample must be from a person who is different from either of these first two. These first two could be from the same person, but this third one must be from a different person because the DNA fragments are in different positions, which means they're different sizes.
Would that be an example of an exclusion?
If you were comparing, suppose this was a known individual and these were two evidence samples. This known individual in the third lane over would be excluded as being a possible contributor to the two evidence samples in the left lane and in the middle lane.
This is done visually as a result of the autoradiograph produced at the end of the RFLP typing process?
it's done visually, and it's also, that's followed up by a more sophisticated measurement. But in fact, the first thing that you do when you get your x-ray film off is you look at it, and you look at the controls, and you look at the samples on there, and you make some immediate judgment about whether it appears that the procedures worked okay, and whether it appears that you have inclusions or exclusions, and based on those inclusions or exclusions, then you might do further testing or you might not.
Following this visual determination, do you then in some instances turn to another process basically to determine whether or not your visual opinion is a correct one as to a match?
Yes.
All right, we'll return to that later. But what I'd like to turn your attention to, Your Honor, I'm going to leave the chart because I don't want to have to ask to bring it up again. And that relates to the amounts of DNA that are in a sample. And I'd like to focus your attention again on this RFLP typing method. If there's no DNA in a particular sample, what do you see at the end of the process on the X-ray or autoradiograph?
If there's no human DNA in a particular sample, at the end, you don't see anything. The lane is blank. There are no bands. It's just empty.
Are there other types of DNA that you could see?
For the procedure as it's used in laboratories for human identification testing, you're specifically looking at, you want to be looking at human DNA, and the test is designed to only detect human DNA. And therefore, if you were using it in a research lab, you could see other types of DNA. But for this purpose, you're only looking for human DNA. If you have no human DNA there, you won't see anything.
So in your laboratory, your interest is in human DNA as opposed to looking at, for instance, animal DNA or other types.
That's right.
What if you have some DNA in a sample? Would you necessarily see results at the end?
If you had some DNA in a sample, you might see some results at the end. There are two measures of whether or not you might see results, and one is quantity and the other is quality.
What do you mean by that distinction?
If your DNA is in good condition, and let's just go back to the spool of thread analogy, if your DNA is unwound off the spool and it's all in one piece, that's good condition. It's like it was in a nice fresh cell that somebody just drew out of your arm. If your DNA is in good condition and you have enough of it, you will get a result.
I didn't know I had my
If
camera
your
on.
DNA
My bad.
is
I'm here.
broken up
I've
in a
been here the
random
whole time.
manner, and the proper term for that is that it may be degraded, you may or may not get a result. And when you think about DNA that's broken up or degraded, you have to think of it sort of as a continuum. It could be only slightly degraded, that is, it's only broken up a little bit, in which case it won't affect your result, you will get a result. Or it can be very, very broken up in very, very small pieces. And if that was the case, you would not be able to get a result with this RFLP test.
What happens with DNA? Does it in fact die or degrade or perish?
Well, I wouldn't use the term die or perish. It doesn't really fit. Degrade is the right term. Or if you want to make that simple, you could just say it's become broken up into many pieces. And that can happen as a result of environmental effects. And you can generally sort of assume that moisture and heat facilitates degradation. That is, it occurs more rapidly, and cold and dry conditions preserve DNA.
If DNA suffers this degradation process, will that result in changing the types from the original type of the sample to a new and different type as a result of the process of degradation?
No,
I'm sorry.
it will not.
So this process of degradation, can it change my DNA into looking like your DNA?
No,
Or your DNA into looking like the court's DNA?
no,
Or any members of the jury or the audience?
no.
Taking you back to this small amount of DNA, what if you have more DNA in a better condition, let's say? Then what do you see?
If you have more DNA and it's in good condition, then you will see bands on your autoradiograph or your autorad. And each time you use a different probe, you'll see bands that probe recognizes. So you can get a lot of information if you have a good amount of DNA and it's in good condition.
Where do you draw the line? In other words, how do you know there's enough DNA there to be able to make an opinion or render an opinion about results from a particular
It
test?
probably takes less time to make a pancake than it does to toast an Eggo.
Well, you can sort of go about it from two directions. You can measure how much human DNA you have before you do the test. and you can run what's commonly referred to as a mini gel, which is a really small gel, about 2 inches by 3 inches, and assess whether the DNA appears to be broken up. So you can measure its quantity and you can get an estimate of its quality. And you could do that before you did the test and then say, well, it looks good, let's do an RFLP test.
Is that, by the way, using the same process of electrophoresis for this mini gel?
Yes, exactly the same.
And that's simply a step to help you evaluate, that is to determine approximately how much DNA is in a sample before you test it?
Yes. If you go back to this green depiction of a gel,
At step 3 on People's 242?
right, if you just made that a lot smaller, and which would then represent your mini gel, you can stain that gel with a dye in which you get a pink smear that looks like these smears that are on this diagram for step 3. And if you had a smear like what's shown in step 3, you'd look at that and you say, well, gosh, I've got DNA sort of spread out all the way from the top of my mini gel down to the bottom. That's pretty degraded. And if it looked like that, you'd be sort of, you wouldn't be sure whether it would be good enough for RFLP or not. If this diagram in step 3 had only shown the pink color at the top, that's sort of what it would look like if it was in good condition and you ran a mini gel. You're looking at a huge piece because you haven't, when you do this mini gel, you haven't cut it with your restriction enzyme yet. So actually, if we could insert an extra step here to be 1A, so that you extracted your DNA and then you ran your little gel and then you made this judgment. If you had DNA that was just hung up at the top of the gel, that would say, you'd say, well, I have very good DNA here. I should be able to go on and do an RFLP test. If the DNA is all spread out such that it looks relatively degraded, then you would have to make a judgment about whether or not you thought it would be good enough to do an RFLP test or you were going to do some other kind of test with it.
As far as these bands, as you see them, and referring to, for instance, step 8 on People's Exhibit 242, those are fairly dark bands in that depiction, correct?
Yes.
You've also held up one autoradiograph, People's Exhibit 246, which also shows a number of bands that seem to be very dark. Is that right?
Yes.
In fact, actually, if you could, with the court's permission, could the witness hold 246 up again?
Yes.
As long as he hangs the towel up this time, that's fine.
To your knowledge, will we return to autorads and
Hang
actually
the
show them in
towel
closer
up.
detail to the
We will.
jury?
Yes, we will.
Okay, but just for the moment, and if you could hold that up. I don't know if you can hold it up without that white background. Let's do that.
Yeah.
Oh, you have Dr. Cotton to step down over there.
All right, that's fine.
She can just hold it up against the white background.
Dr. Cotton, again, showing People's Exhibit 246. That shows a number of very dark patterns. Is that right?
Yes,
Or bands, is that the appropriate term?
it is.
Now, with regard to samples, for instance, that you receive in your laboratory, will the darkness or the intensity of those bands vary?
Yes.
Why?
The darker the band, that reflects you've had more probe bound to it. More probe means that you had more DNA there. So if we just we don't have anything on here that has very light bands, but if you had something that had very light bands, that would be an indication that you didn't have very much DNA. If you have bands that are darker, that means you have more DNA. Now, there's other ways you can make the length of time that you lay the X-ray film over the nylon. The longer you lay it on there, the darker everything becomes, but it will still be relative. A darker band generally has more DNA than a lighter band. And there's a few reasons why that. There's a few technical exceptions to that, but that's the correct generalization.
All right, then if you would have a seat back on the witness stand. Is it the case then, Dr. Cotton, that with regard to samples with not much DNA in them, but enough to produce a banding pattern, you may see bands that are faint?
That's exactly right.
Is faint a term that you use in your laboratory?
Yes.
What does it mean?
It means that instead of a dark, crisp line, you have a difference between where there's no band and where there's a band, but the level of darkness there, that is the intensity, is much less. It's light.
Does that have any significance in your interpretation of results?
You are always looking at the position of the band. So whether it's dark or it's light, it's the position that you're interested in. It does affect, that is, you think about it in your interpretation because you're assessing, I have a lot of DNA here, I don't have very much here. Um, and you may use that information in your interpretation, but it's mainly the position that you're interested in looking at.
What about in terms of faint bands and whether or not you can see them? Does that play a role? I assume that plays a role in your interpretation.
Well, if you want to interpret them, you better be able to see them.
Are there any policies in effect in the laboratory at interpreting, for instance, results that have faint bands?
I don't believe that we have a specific written policy. We do have a common practice in our lab in dealing with faint bands, and we do follow that practice generally whenever we encounter faint bands. It's not actually a written part of our procedure.
And what is that procedure, though not written?
There are two parts to it. In the further analysis that we haven't really discussed yet, which involves a computer imaging step, there is a standard level of darkness that we ask that the computer see in order to call something a band. In addition to that, we have to be able to see it ourselves. That is, both the computer has to be able to see it and the analyst has to be able to see it. And if we really are struggling with whether or not a band is light, we will ask several people to come in and cover up the adjacent lanes and make an assessment as to whether all the people involved in doing the analysis of the test agree that they can see a particular band.
When you say cover up the adjacent lanes, is that to ensure that whoever's looking at it wouldn't be biased by the presence of bands in other samples?
That's right. If I'm working on something or another PhD staff or one of the analysts is working on it, if I'm not sure whether or not something is intense enough for me to use it further in the interpretation, I might lay a piece of cardboard down on either side and just say to one of my coworkers, would you come in and look at this and tell me where you see bands in this lane? We don't always do that, but that's generally what we do when they get quite light.
As far as this process of DNA, once it's degrading, does this degradation process ever lead to bands appearing that weren't in the original DNA in the sample?
No, it doesn't.
So in other words, you either see the DNA from the original sample, or if the degradation is too great, you don't see anything at some point.
Well, you've just said what the extremes are. You may have an intermediate result where you have the DNA is somewhat degraded, and the effect that that has is you tend to lose the bands that are larger, that is at the top of the gel. So you may end up with a partial set of bands. You don't see the ones that are large, but you see the ones that are smaller in size. And that can happen also. What won't happen is that with degradation, you won't have a band at one size and then have it converted to now a new band at a different size. That's not going to happen.
Okay, and perhaps lastly for today, with regard to this computer imaging process that you just described, can you tell us more about what that is?
It's sort of like this Elmo machine, actually. Well, kind of. Um, the computer imaging system consists of a light box and on which you lay your film, a camera at the top, and a computer monitor so that the camera is capturing the image of the autorad and displaying it on the computer screen for you. And then you're asking the computer to help you do some manipulations in terms of saying, here's a band and here's a band and here's a band. And then the computer will also, because of its software, do a calculation for you that says the band in this position is approximately 5,000 base pairs.
Now, when you say the autorad that you have the camera actually focus on, is that an autorad just like People's Exhibit 246?
Yes.
And then this camera and this software in combination then review the results on that particular autorad?
Well, the reviewing makes it sound like it's thinking about it, and it's not. What it's doing, it's visualizing. It's a video imaging system. It's taking a picture of that autorad and displaying it on the computer monitor, and then you are directing the machine to do particular things, not to the film itself, but to the image.
And what are those things that you direct it to do?
You're telling it where the control lanes are. You're telling it where the sample lanes are. And you're telling it which lanes you want it to do a calculation to find a band. That is, the computer will basically scan down
They
and where
can't.
there's a dark.
They just can't. Let's just see if we can see. Can we just see if we can see how long we're going to have to wait to figure that out? Can't tell, can't tell, can't tell, can't tell. Okay, it's fine.
area, it will identify that as a band. And where there's, so it's looking for changes in levels of darkness. So if it's clear and then it sees a dark area, then it identifies that as a band, and then it sort of scans down and finds another band and identifies that. So it's making an identification of a band position, and it can do this correctly and it can do it wrong, and the operator has to help it along. And then once you're happy that it's identified bands and you agree that those are bands, it will do this calculation for you that will give you an estimate of the size of that DNA in base pairs.
And that's the purpose of this imaging process, to assist you in interpreting results. Is that right?
That's right.
And also to establish the sizes for the various bands according to the methods that you just described?
That's right.
Good spot.
Yes, thank you, Your Honor.
All right, ladies and gentlemen, we're going to take our recess for the afternoon. Please remember all of my admonitions to you. Don't discuss the case amongst yourselves. Don't form any opinions about the case. Do not conduct any deliberations till the matter has been submitted to you. And do not allow anybody to communicate with you with regard to the case. As far as the jury is concerned, we'll stand in recess until 9 o'clock. And we have a hearing tomorrow morning set at 8 with Mr. Ullman and I don't recollect who from the prosecution. Photographs tomorrow, 8 o'clock.
We should
All right, we'll stand in
deliberate.
recess. And Dr.
We
Cotton,
should
tomorrow
discuss the
morning,
case
8:45.
and
All right,
deliberate.
thank you.
What are you going to do, Ida? What are you going to do? TG.
All right, good morning again, Dr. Cotton. You reminded, ma'am, you were still under oath, and Mr. Clark, you may continue with your direct examination.
Thank you, Your Honor. Good morning, ladies and gentlemen. Dr. Cotton, as far as these methods of DNA typing, and you've talked in broad terms about PCR and in more detail about RFLP, are these techniques simply a better way to type stains than, for instance, ABO typing or conventional serology?
Yes, they generally will offer more information than conventional serology.
Contraband.
Again, we spoke about RFLP, or you spoke about RFLP typing yesterday. The second technology that you mentioned briefly yesterday, I believe you referred to as PCR. Is that right?
That's right.
What year was that process discovered?
I believe that the first paper on PCR was published in 1985.
As far as its use in forensics, is it newer or older than the RFLP typing process?
It's actually it came along at about the same time, although it was not being used quite as widely early on as the RFLP process.
Are you familiar with who was the first scientist to use this PCR process in forensics?
Yes, I am.
Who was that?
That would be Dr. Ed Blake.
Do you know approximately when that forensic use of that technique occurred?
I only know that it was early, maybe 1986 or so.
As far as this PCR process, and many of us, not all of us, have heard of the movie Jurassic Park. What does PCR have to do with that?
Objection, Your Honor.
Sustained.
PCR does what? Can you give us a very brief summary?
PCR is a way of saying, I want to replicate a small section of DNA, and it allows you to replicate a small section of DNA in a way that if you have a very small amount of starting material, you can go through a sufficient number of.
Yep, they objected to Jurassic Park, and I'm not surprised. Poor Alan is going to miss this riveting murder testimony.
Replications, so that by the end of the process, you will have enough of this small section to then do some analysis on.
What role does it play, for instance, in old samples? And let's first of all ask, can this process be used on samples of DNA that are perhaps years or decades or centuries old?
It can, and it is being used on those types of samples.
What types of samples is that?
There are samples from, I don't know what the proper term would be, but very ancient, for example, bone samples that have had very short sections copied using PCR to look at how genes have changed over time. That is, if you can have a sample from a human being who is several thousand years old and look at a particular sequence of the DNA, you might say, well, how does that sequence compare to the same sequence in modern human beings?
Has PCR been used on ancient animals?
I don't really know. It may have been, but I don't really know.
As far as this PCR process, is it like RFLP in the sense that it's a testing method, or is it something a little bit different?
PCR is simply the process that allows you to make a lot of copies of a particular section of DNA. And then the rest of the analysis may have many different forms. That is what you do now with your section of DNA that you've replicated many times, may not be the same for every purpose or every question.
Would it be correct then that PCR would be more like one step in a typing process when DNA is being typed?
Yes.
What about your own knowledge of PCR? How did that come about?
I had been to several meetings where PCR was discussed. I had done some reading where PCR was discussed. My own hands-on experience came from visiting 2 different laboratories where they were actively doing PCR and learning from the staff in those laboratories how to do PCR. And both of these visits to other laboratories, one was 2 weeks and one was a week. I had an opportunity then to learn from the staff there, to do it myself, and then going back to Cellmark to help the rest of the Cellmark staff, some of whom had also been out to get training, which was different than mine, to set up the PCR that we do there.
Who were the individuals that whose labs you visited to learn PCR?
I went to a laboratory at Houston. That was Dr. Tom Caskey. And I went to Dr. Alec Jeffrey's lab in the UK.
The same Dr. Jeffries that you described yesterday?
Yes.
How about your own personal hands-on experience with PCR? Could you describe that, please?
Well, I had those 3 weeks, and then I had some experience, hands-on time doing the PCR reaction while we were in the process of doing validation studies to set up PCR in our laboratory, and a number of other people did too. I am not doing the PCR test now. The PCR tests are done by the regular lab staff.
As far as this copying process, is there a term used to describe what you actually do in taking small segments of DNA and copying them over and over?
Yes, the term is amplification. So people will talk about, I've amplified a certain section of DNA.
This process, and I believe you said that this process of copying over and over is directed at short segments of DNA?
The segments that are used for forensic analysis are relatively short, and I'm talking about perhaps 100, 200, up to 400 base pairs in length. That's in DNA terms, that's a pretty small piece.
So you're not talking about copying over and over this entire segment of DNA that you described as containing, 6 billion bases or so?
No, you're not copying the whole thing. You're just copying a particular section.
With regard to this copying process, would it help to use a chart or a pad so that you could draw what this amplification or copying process involves?
Sure.
All right. Your Honor, then with the court's permission, Mr. Fairtlow will, I believe, set up the drawing pad for the witness.
Reassemble our drawing pad.
Reassemble.
Yes. And Mr. Clark, I noticed yesterday that you were using the dry erase type marker. Does that have a longevity to it, permanence to it?
I hope it's not disappearing ink.
You notice that's what you're using there is dry erase stuff.
One over there. The dark blue one, I think, is just a different.
This diagram be marked as people's next in order, which I believe is 247.
247.
All right, Dr. Cotton, if you could step down from the witness stand. And what would be an appropriate label for this chart to aid you in describing this copying process or amplification?
We'll just call it amplification.
Very good. If you could just write that at the top. All right, now if you would go ahead and if you could describe for the jury, please, what is this copying process?
Okay, keep in mind now that you're starting at have a sample and you've extracted the DNA. So those steps are the same as or similar to the RFLP process. And so let's just say, for simplicity purposes, that we have a single chromosome's worth of DNA that I'm going to use as a reference point here. So we're going to put in the tube our starting DNA. And we're going to put in the tube a supply of the 4 DNA components: A's, T's, G's, and C's. And we will also put in the tube an enzyme. This is a protein, and it's called a polymerase. And this polymerase is basically going to assemble the A's, T's, G's, and C's. And we're going to put one more thing in, which is referred to as a primer. And that's a very small piece of single-stranded DNA, usually about 20 bases or so. It can be longer or shorter, but that's just an example. So that we now have the components to make more DNA. This is what we're going to use to make more. We need our starting material. We need the building blocks. We need something to put the building blocks together, which is the polymerase. And the primer serves as an anchor for that polymerase to hold on to.
Let me stop you for just a moment, Dr. Cotton. With regard to this term polymerase, is that the first word of PCR, the process itself?
Yes.
And what does it stand for again?
Polymerase is a protein which can assemble more DNA in this case.
And what does PCR actually stand for? What are the words?
The PCR stands for the polymerase chain reaction. And basically,
I don't think so.
steps, which is
I'm guessing, and I don't know, that they have a lawyer that's more versed in DNA that is going to try to question her. And I'm dying to know who it's going to be.
why it's referred to as a chain reaction. You're repeating a cycle of steps over and over and over again. And what we'll do here is just show you what 1 or 2 cycles looks like.
All right, go ahead.
So the first thing we'll do is heat this DNA back up to about 95 degrees, enough so the strands will separate.
Again, is that centigrade, not Fahrenheit?
Yes. And at that point, we'll allow the short primers or anchor points to bind, and these primers are designed so that the sequence of DNA that they have to hook up to is known, and so you have a single-stranded piece in the primer binding to this now single-stranded starting material, and all the A's and T's and G's and C's are pairing up. And then the polymerase will come along and, directed by the bases on your starting material, will reassemble a second strand.
We haven't seen this lawyer for the prosecutor, or we did way, way, way at the beginning.
I suspect we have a vision
Maybe.
problem.
Yes.
Preliminary.
Or, Your Honor, would it be possible to put the drawing pad here where the podium is?
Let's put it there. Why don't you turn it as directly perpendicular as you can? Thank you.
I'm not sure we solved all the vision, though.
All right, juror number 1, can you see that? All right, thank you, counsel. Proceed.
And if you could.
I'm
would you
completely
briefly
against
describe
delayed
again what you've already
gratification.
described, and point to the
Give
various
me it.
portions
Give
of
me
the
it
chart so
now.
that each of the jurors may see it.
We have double-stranded DNA starting material. We're going to heat, and the 2 strands, we'll just put a little X here on our starting material strands. Drop the temperature down, allowing the primer to bind, the 2 primers to bind, and then allowing the polymerase to make a copy of this starting strand. So the polymerase is basically going along and says, I have a T on my starting strand, I'll put an A in. I have a C on my starting strand, I'll put a G in. I'll have a G on my starting strand, I have to put a C in, and so on. So now where we originally had a single strand to start out with, we now have 2. Now
Go ahead.
I'm sort of going to run out of a length of paper, but the idea is that you then start the process all over again. So you go, stand this up. So you go, start the cycle again, heat it up. These two double strands will come apart. These two double strands will come apart. And now again, you drop the temperature down and let the primers bind. I've just made these shorter because there's not enough room. They wouldn't really be shorter. They'd be the same length. And then allow the polymerase to finish off making the copy for each one of those. And so now we have 4 completed strands where we only started with 1. So as you can see, each time you go through the
No,
cycle,
no,
you're going to
no.
double.
That looks like a pyramid scheme, and I hear they're bad. So, no, no, I'm staying away from DNA. It's going to take all my money,
The number
all
of
my
strands that we
friends.
started out with. If we start out with 1, then we would go to 2, and then we would go to 4. The next cycle, you would go to 8, and so on.
All right, perhaps you can stand on the other side so that I can ask you a question. A few questions about this process. You've used the term primer. Does it act as something like a primer in priming a pump, for instance?
Well, it's not a bad analogy. It's not great. What it is the characteristics of the enzyme are such that it needs an end to start from. If you just separated the strands, it wouldn't have a place. It doesn't have an anchor point. So the primer is serving as an anchor point for the enzyme to then connect the next base to. It needs something in the way of the start of second strand to actually connect. So the primers are in there both to say this is the section we want to copy. That's really what the primer does, is it identifies the section that you want to copy, and then it serves as an anchor point for that enzyme to sit down and then move through and copy that piece of DNA.
How do you know where the primer actually attaches to this segment of DNA?
You have designed your system. That is, you have to know up front the sequence of bases of the piece of DNA that you want to copy. There has to have been enough research done and development done so that, at least in the forensic setting here, you must know ahead of time the sequence of bases that exist in the piece that you want to copy. That has to be done ahead of time. Then you can manufacture in the lab the primers that have the sequence at the beginning and the end, and then you're set to go.
As far as these known sequences of where it starts, for instance, and you mentioned you have to know the sequence, have scientists looked at these areas to determine their sequences?
Yes, the development work for the genetic locations that are used for forensic testing with the PCR methodology, these regions have all been sequenced, and a lot of other work besides the sequencing has been done to study these regions before applying that information to actually designing a forensic test.
This copying process, first of all, do our bodies do that by themselves?
Not with this particular polymerase, because this polymerase is able to work at a very high temperature. But all our bodies have a similar enzyme, so that when a cell replicates and divides to form 2 new cells, in the process of doing that, you are replicating the DNA, and the enzymes that perform that replication are also called polymerases.
Would it be correct to say then that this PCR process simply does outside the body what the body does on its own?
Basically, that's right.
And it's as simple as that? In terms of this division and copying and
Yes.
so forth. All right. Now, Your Honor, if we could, I'd like to put that drawing pad back where we had it yesterday and then continue on with some additional prepared charts. All right. And perhaps you can have a seat again on the witness stand, Dr. Cotton, but probably not for too long.
That's not so good. I can't see either.
Now, as far as this use of PCR, and I'll step over here, Dr. Cotton, as far as this use of PCR in forensics, to your knowledge, do we have some charts, prepared charts that illustrate how you actually use this technique to type samples in forensic casework?
Yes, I've seen the charts that you're referring to.
All right, Your Honor, at this time, I'd ask to be marked as People's Next, which I believe is 248. For the record, this diagram can be labeled or can be described as PCR analysis, and then showing at the top 2 large blocks, step 1 extraction and step 2 amplification, and then a smaller block labeled step 3 detection.
Yes.
All right, Dr. Cotton, with regard to this chart that's been placed on the board, have you had an opportunity to look at this before this morning?
Yes, I have.
And with regard to this chart, does it illustrate various steps in the PCR-based typing process in forensics?
Yes, it does.
Now, in particular, well, are there a number of steps that occur in this typing process?
For sure.
In general terms, are there fewer steps than in the RFLP typing process that you described yesterday?
Oh, there might be fewer. It sort of depends on how you count them.
Okay. This board itself, and it's labeled at the top, step 1 extraction. Can you describe what that refers to? And if there's a pointer there that would help you, please feel free to use it.
The top rectangle is simply illustrating that you have some kind of stain, and from that you do a DNA extraction, and the DNA is going to end up basically in your test tube. And these tubes are actually very small. And then to that tube, you will add, in addition to adding your DNA, you'll add what's diagrammed up here as the PCR mix. And the PCR mix is the DNA components, the A's, T's, G's, and C's, the polymerase, and the primers, and some other salts or buffers. So all of those things go together in a single tube.
All right. What happens at that point?
At that point, you take that tube and put it in what's called a thermal cycler. It sort of looks like a cash register, actually, or a small one, but it has a metal block, which is illustrated right here, and a computerized, programmable more control.
Remember the plain old thermocyclers? Mine now is bedazzled. It's all pink and glittery. My thermocycler that I have here to, cycle my thermos after dinner.
Controlling the temperature in that metal block. So the tube sits down very tightly. The holes in the block are basically the same shape as the tube, so that when you put the tube down there, there's close contact between the tube and the walls of the metal block. And the temperature in the tube is then controlled by heating or cooling the metal block, and you do this in a cyclical manner. And the cycle goes, as we talked about a little while ago, you're heating the block up to 95, so the contents of the tube will be at 95, so you can denature the DNA, separate the strands. The temperature is then dropped down, and I'm not going to be able to remember what the exact temperatures are in the cycle, but the temperature is dropped down somewhere usually around 60 or 64.
My
65
doggie's
degrees.
crying. I don't know why.
Could even be lower than that. Anyway, I don't remember right now. To allow the primers to bind. And then the
I'm going to go
temperature is
give her
raised
some
up
chicken.
to around
Be right
70
back.
degrees to allow the enzyme to complete copying of the strand. And then the cycle starts all over again. So you tell the machine in your programming what specific temperatures you want it to cycle through.
I see why she's
how many
crying.
cycles you want it to go
I
through.
figured it out.
In other words, this machine, you can program it or tell it basically how many of these cycles to go through, at what temperatures, and so forth.
That's right. And how long each cycle.
Yep. You can program it to tell it how many cycles to go through. My dog's crying because it's been 2 hours and 10 minutes now. She knows. She's like, yo, you're done. Get off the computer. Get off the computer. Yes, dear. All right, guys. We did it. We're the whole way to March 9th. Yay. And now we know who's coming next, but hopefully, well, I can't say hopefully we'll get through it quick because we all know we're not. We all know that even though we don't know who's here, she's back down here. She's back down here. She's back down here. So there's no use even looking. We're going to be here for a long time. Have a great day. I'll see you tomorrow. Behave. Don't join any multi-level marketing schemes today. Bye, guys. 1995, May 9th, 1995. That's where we're at. I think June is when Furman comes back on. I think. Anyway. Bye, guys.